Hi everyone, welcome back to video 3. This time we are just going to discuss cancer and how cancer develops from an uncontrolled cell cycle. So in video 2, when we talked about mitosis, I started off by mentioning that mitosis is a highly regulated process. There are checkpoints that say that the cell is okay to divide or that the cell is not okay to divide. And if it has the message that it's not okay to divide for any reason whatsoever, it goes through this programmed cell death called apoptosis. Now, that is a normal part to maintaining healthy cells and a healthy organism. Again, you don't want a cell that is too old or too damaged or one that has acquired too many mutations to be allowed to copy those mutations and pass them on to future cell lines. You want to stop them dead in their tracks. Now, cancer cells are cells that don't follow those regulations. They don't listen to the checkpoints. They grow and divide as they wish, and they have acquired multiple mutations. Now, I like dissecting how cancer cells work. I think because when you take a look at all of the things that have to go wrong for cancer to take place, it makes you appreciate all the things that cells get right 99% of the time. And I think I've expressed this to you when in our face-to-face classes that it really is fascinating that you have these cells that just with chemical signals can do so much, and they do it with such accuracy um, that it's quite amazing. So let's take a look at cancer cells here. So cancer cells are uncontrolled cell growth. Again, they no longer listen to the checkpoints. They just grow and divide, grow and divide, grow and divide, and they do it at an unregulated rate. So what's happened? Okay. So number one, cancer cells don't follow the rules. There are a couple of things called the hallmarks of cancer. And over here, I've listed a couple of them. And now, as research gets better and scientists spend more and more time studying cancer cells and they study the genetics of cancer cells, they're finding more and more of these hallmarks. But a couple of things that they do is number one, they have the ability to steal nutrients away from healthy cells. How do they do this? By a process called angiogenesis. That means that they can create their own blood supply. So over here in the diagram, I have a tumor that's growing. Those are the cells in blue. So imagine these cells in my tumor have the ability to grow blood vessels and have those blood vessels reach and merge with the blood vessels of healthy cells. And now what happens is that all of the nutrients that should have been going to the healthy tissue start feeding the tumor instead. In fact, one of the new cancer tools that they're looking at to destroy tumors is to create medicine, to create drugs that can stop the process of cancer angiogenesis. Because if you can starve the tumor, you're going to cause the cells to shrink. The other thing that cancer cells can do is detach from their origin. So you can have, and that's what's going on in the picture actually, you can have a tumor and you can have one single cell uh, break off from that tumor and find a ride, hitch a ride into that blood vessel that it created. And now, once it's in the bloodstream, it's free to travel and set up a new tumor wherever it lands. We name cancers after the um, first tumor origin. So, someone who has lung cancer, we call it lung cancer because that's where the tumors were first diagnosed. But somebody who has lung cancer, I'm sure you know, may also develop cancerous tumors in their liver or their brain, in their bone. So that was all because of metastasis, because some cancer cells from that original tumor broke off, found their way to the bloodstream, and now they've set up shop someplace else in the body. The other thing that cancer cells can do is grow in multiple layers and dimensions. So typically your cells grow in one layer, right? Think about when you get a cut in your skin. The skin cells that are surrounding that cut, they get a message that, hey, there's a gap here in this organism. We need to send out the signals to grow, to repair the wound. And once all of the cells recognize that they have a neighbor again, they're going to get a signal to stop growing, stop dividing. However, cancer cells don't obey that rule, and they grow in multiple layers, in multiple dimensions. And that's why you get those weird, irregular shaped tumors because they don't follow that layer and dimensional rule of healthy cells. They acquire mutations, so they don't go through those checkpoints of was all the DNA copied correctly. They don't care about the mutations. There's mutations made during DNA replication. There's chromosome mutations made where parts of the chromosome break off or they duplicate themselves. We'll talk about those things later. But they don't ever go back and fix any of these mutations. They don't go through any of the programmed cell death, and that's what we're getting to here. They remain immortal. They have the ability to continuously divide their DNA, and they also avoid that apoptosis, that programmed cell death. And I have down here a special line of cancer cells, HeLa cells. We'll talk about more in a minute. So on the next slide, I just sort of found this graphic of some of those hallmarks again, right? The fact that they resist cell death, they resist that apoptosis, they can make their own blood vessels, angiogenesis, they have genome instability. That means they acquire all of those mutations. They can have, more than the correct set of chromosomes, etc. They can invade and metastasize to other parts of the body. And up here, this cell signaling and growth suppression. So that's what I'm going to talk about next. And notice that in each one of these hallmarks, you can see it says 7 genes, 16 genes, 15 genes, etc. This just identifies the number of genes that scientists have discovered that play a role in this particular hallmark of cancer. And so you can see there are lots of genes that cancer cells mutate and change that are really regulated to avoiding apoptosis. There's a lot more that they have acquired mutations in their ability to, obey those stop and go signals of cell division. So how does this all work? So let me preface by saying, in all of our genomes, you and I included, in all human genomes, there are tumor suppressor genes and there are proto-oncogenes. This is part of our natural genetic makeup. Now, tumor suppressor genes, in a healthy cell, the job of a tumor suppressor gene is to tell cells to stop dividing. That's what suppressor means, right? You are suppressing division. So a tumor suppressor gene should tell a cell that, hey, you don't need to divide anymore, or you're too damaged. I don't want you to ever divide again. You should go through apoptosis. Imagine, let's equate this to the cell cycle or a cell going through division to operating a car, to keeping a car functional, right? And we know that to make a car, to keep it functional, to keep it moving, it's a balance between the gas pedal and the brake pedal. So in a tumor suppressor gene, that's going to be our brake pedal. The car is going or the cell is going, it's dividing, it's going down the road of cell division. And the job of the tumor suppressor gene is to put the brakes on when appropriate. In a cancer cell, however, tumor suppressor genes are mutated and they're sometimes deactivated, meaning they're turned off. The cancer cell doesn't even listen to it anymore. It's completely shut the light off. In that case, the cancer cell doesn't stop dividing despite getting the signals to do so. It's almost like cutting the brake line to the car. And so it just keeps rolling on down that hill. Now on the flip side, you have proto-oncogenes. And again, proto-oncogenes are a gene that we all have in our genome. And in healthy cells, the proto-oncogene is like the gas pedal to the car. It gives the signal to the cell to properly divide. That yes, it's okay to divide, go. Yes, you're okay to divide, go. Now, in a cancer cell, proto-oncogenes become mutated. And when that happens, they're called oncogenes. An oncogene is a mutated proto-oncogene. And what an oncogene does is just keeps sending out that go signal. It just keeps telling the cancer cell to divide, 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 and do it quickly and fast, as much as you can. And when you've got a cell growing that quickly, repeatedly, it's not going to stop and check to see if there are mutations being made, if there are chromosomes missing, if all of the chromosomes are attached to spindle fibers. So that's how these cancer cells start to acquire mutation after mutation after mutation, and they get to what is called that genome instability. So the proto-oncogene, or once it's mutated and it becomes that oncogene, the oncogene would be like the gas pedal that's stuck to the floor. And it's just telling that cell to divide, divide, divide. Now, I mentioned HeLa cells. So we'll wrap up here talking about HeLa cells. And so HeLa cells were cells that were taken in 1951 from a African American woman in Virginia. And she was fairly uneducated. I think she had an 8th grade education. And I say that because I don't think that allowed her to question what was going on at the time as best as she could. But Henrietta, she was young, in her early 30s, and she had just had her 5th child, and she was experiencing some prolonged bleeding after leaving the hospital after having her 5th child. So she went to Johns Hopkins Hospital, and a doctor there by the name of Dr. Guy did a pelvic exam on her and found that she had a cervical tumor. So, without her permission and without her knowing, he took a biopsy of this tumor. Now, a little side note about Dr. Guy. Dr. Guy, for a long time before he met Henrietta Lacks, was trying to get cells to grow in culture in a lab, meaning, getting cells to grow outside of the human body. And he was having a really hard time doing this. It wasn't working for him. So he took this biopsy of Henrietta's cancer and he cultured it, put it in a petri dish, gave it some nutrients, and it just took off. Her cells from her tumor just grew and grew and grew and proliferated. And Dr. Guy successfully became the first scientist to get cells to grow in culture in a lab outside of the human body. Now, they were given the name HeLa cells because at that time you named cells samples from the first 2 letters of the first name and the first 2 letters of the last name. So Henrietta Lacks, we have HeLa cells. Now, HeLa cells are still alive today. Um, Cell lines from her original biopsy cells continue to grow in labs all over the world. They are sold in biomedical supply books. If you're a lab, you can purchase them. They have been used to discover the polio vaccine, the human papillomavirus vaccines. They have been sent into space to learn what the lack of gravity does to cells. Her cells have been so crucial to numerous scientific advancements and discoveries, not just in the United States, but again, all over the globe. All right, so how does a cell acquire some of these mutations in the first place that could possibly lead um, to cancer? So number one, we can have damage to the genes, right? Like I said before, damage to these tumor suppressor genes. An example of that is the P53 gene. But a lot of times it's exposure to carcinogens, which are materials that we know cause cancer, things like heavy metals, toxins, asbestos, and then we can have things that we know are mutagens. Mutagens are materials that can cause mutations to DNA, but they don't necessarily lead to cancer. I mean, they probably do. There's a high likelihood that they do, but they don't necessarily have to. So UV light, clearly we know can lead to skin cancer, but it's not always the case. Tobacco use we know can lead to lung cancer. If it's inhaled into the lungs, if it's a chewing tobacco, it can lead to cancer of the jaw and the gums. Um, but again, not always the case, right? So you can see there are just some examples of other things that can cause cancer, viruses. In fact, viruses like Epstein-Barr virus, which is the virus that causes mono, human papillomavirus, we know has a link to cervical cancers. The Epstein-Barr virus has a link to Hodgkin's disease. Sunlight we know can cause skin cancer. tobacco use, diet. Notice how diet makes up a huge part of this. Okay. I really am a believer that food is medicine, and eating a wide variety of good, healthy, unprocessed foods um, definitely helps support the maintenance of not just healthy cells, but also healthy DNA genetic material. All right, so you have an activity to learn more about HeLa cells. So I didn't go over that too much in depth, but I am going to give you a couple of exercises in your assignments to learn more about that. All right, so there's our story on cancer, and until next time.